Welcome. In this video I'm going to try to review how to use the GLX to do our temperature recording, and then how to go back through the valve procedure, and then briefly how to finish a trial. Okay, so to get started, we're told to measure temperature to the nearest 0.01 degrees. You see how that's going to the 0.001 degrees? We can change this by going to the home menu and then going to the sensors. So we'll arrow over to sensors, which is right here. So sensors, and hit the play button, or no, the check mark to get in there. So we use the check mark buttons like the OK button. And then, it's like dark right here. And then we can change the sample rate. And right now the sample rate's set to 2 hertz. And so we want to change that to seconds and set the sample rate to 2 seconds. Okay, I don't know if we can see this here or not, but sample rate to seconds and then the 2 seconds. So it's going to record a data point every 2 seconds instead of 2 per second. And then what we want to do is go all the way down to temperature. So we just arrow down to temperature, and then we want to hit F2 for properties. And then if you see here, the number of fixed decimal places right now says 3. So the number of digits, we want to have that say 2. Okay, and so what I'm doing is just using the plus and minus buttons down here to either add or take away a decimal place. So we're going to 2, I can hardly see that, maybe you can see it, but 2 digits. Okay, and then to save, we got to hit OK. Let's hit F1. And then once we're done here, we can just hit the home button to go back to the home. And then we go over to table, to go to our table, check mark button. And then if you don't see temperature and time here, what you would do is use the arrow buttons to arrow. Let's see, okay, we can use the arrow buttons to arrow over. So like if you want to go back to an old run, you would check mark on run 14, and you could go back to an earlier run. So if we click on run 14, we could go to an earlier run. I really don't want to do that, but if you want to go back to an earlier run, you can do that. You can use the escape button to go back, to exit out of that menu, but just hit the check mark button again. Temperature, if we want to change the axes to be opposite, this is where we can put temperature, time, or other properties into one of the columns. That looks good. I'm going to hit the check mark, and then I arrow over to time, and it's time there too. And if this is like temperature versus pH, we just change the pH cell over here to be time. And then all I need to do is hit play, and it should start taking data with the proper settings. I may have actually been taking data the whole time here. Let me hit play again. Okay. So there we go. It's still going to 3 decimal places, not 2. That's not the end of the world, of course. Let me hit play. So the play button is to go and to stop. So it's like the play and the stop button. Once you stop and restart, it would start a new run. So it's not going to continue on your run. Another thing we can do is get into the properties. Like I can go over to temperature here, and then I can hit the OK button and still get into properties here. So I can get into properties from the sensors menu. I can also get into the properties. I think I can get into it here. But in any event, if we want to change those properties, we would just go to our home screen, back over to sensors. I think I was actually doing a run while I was trying to change the temperature settings, which is probably why they didn't stick. Well, it should be 2 decimal places. I don't know why it's recording to 3, but more is better than less, so I'm not going to be too worried about collecting an extra decimal place on our temperature readings. gonna go ahead, stop this, and start a new video. Okay, so here we are. I got time running here, and it's actually collecting to the nearest point the nearest point 0.01 degrees C like we want. The only thing I might add here is what you want to do is when you're doing your valve procedure and you close valve 1, you open valve 2, and you're like watching this pressure drop below about 100, that's when you want to hit play. There's going to be some error no matter when you hit play anyways. We work about 10 to 20 seconds of error into our time calculation because it's hard to pinpoint at what point the sample begins to start escaping from the cell. Because what we need to do is drop the pressure so that the compound as it's supplying the molecule doesn't bounce into a molecule of air. So that's really why we need the vacuum system here, is just to lower the pressure, the surrounding pressure, so the mean free path is long enough for the sample to actually escape from the cell and be pumped away by the pumping system. Okay, and so we're kind of guesstimating that to be about 100 millitorrs when that happens, which is when we hit play here. And then what we're gonna do is we can track time right on the GLX throughout the experiment. You might wanna have a stopwatch going just in case something goes awry, but it's gonna be tracking the temperature on our water bath. Now, if you notice, the water bath temperature, like I had set it to 35 degrees and it's still warming up, it's only to 34.3. What I probably would do here before I start the run is just match the set point. Because the whole idea here is to try to keep a constant temperature throughout the trial as best as possible. So if we just match the set point to the current reading of the temperature, might be the best way to go about doing that. And then the other thing is that usually when we're monitoring the temperature, we want to, place the thermometer. We could use tape or dangle it in such a way that the cell, and of course, like I haven't even submerged the cell in the water bath, but you'd want to, of course, make sure you raise the water bath up to submerge the sample. But what you want to do is track the temperature in the middle of the water bath near the sample. And if we look at the temperature that's being recorded, it's about a degree cooler than the water bath says the temperature is, which is pretty much what we tend to see. So the water is usually a little bit cooler in the middle of the bath than at the bottom of the base, which is where the water bath is actually reading and monitoring the temperature. Okay, so match the set point, get your thermometer set up, be prepared then to close valve 1, open valve 2, watch for that pressure to lower, and then as soon as it's lower than about 100 millitorr, hit play. It only takes maybe 5 or 10 seconds for that pressure to lower. We're assuming about 10 to 20 seconds of error in time, so the time's not super crucial. So don't be too stressed out because you got a lot of things you're trying to watch and do at the same time. And then at that point, you can close 2 back up, reopen valve 1. So to go back through, close 1, open 2, watch the pressure, hit play, close this back up, open this back up, and then open valve 3. At this point, I haven't stopped from my previous video, so we're at about 6 millitorr versus 26 millitorr in the exhaust line. So we're getting a little bit of a lower pressure in our sublimation region as we expect for our trial. And so then the specific time you run for is also not crucial. What's crucial is that you monitor the time and be accurate with how long the actual trial went for. So you can run a trial for 39 minutes, the next one can go for 46 minutes, it doesn't matter as long as you actually track the specific time your trial runs for. And the way I would end the trial, it's actually very simple, is you just close this valve here. And then as long as you hadn't made a mistake earlier, this valve's closed here. If this valve's open, we got a problem. But this valve should be closed. So just close here, double check the back valve 1 is closed. Keep this open. Some groups start shutting everything. Keep this open, keep the always open open. This valve here between the diffusion pump and the exhaust pump, we're only closing this during the pre-vacuuming step. So during our post-trial, we just close here, relatively quickly, go ahead and hit stop here and then remove your cell. Don't hit stop and then wait 5 minutes because the sample may continue to sublime and leave the cell. So then to remove, all we do is take this off, well, lower the water bath, which it already is lowered, but lower the water bath, start taking this clamp off here, use 2 hands of course, and just break the seal, just break it this way. You hear a hiss. If you hear the pump working, if this pump starts making a loud noise, something's wrong. Some valve is open, so try to find the valve. It's probably this one, could be this one if you forgot to close that one too. But we gotta make sure that we're not pumping on air because that's bad for, in particular, diffusion pumps. Okay, and then we would just go back to having the cell, and then those two clips on top of it, and go take that final mask. Okay, well, another step would be making sure the cell is dry. So you want to remove it from the bath, get it good and dry, and then go take that final mask. So you want to try to dry the cell off before taking the lower clip off here. So remove this clip here, get the whole cell dried, use a whole lot of paper towels, and then take that final mask.